Like several of the academic staff of the University of Sydney, my research concerns coral reefs. I'm interested in the ecology of reef fish communities. The data which I've been entering here were collected recently at Sydney University's field station at One Tree Reef. Lying in the center of the Capricornia section of the Great Barrier Reef Marine Park, at the southern end of Australia's magnificent Great Barrier Reef, is One Tree Island and its surrounding reef. Like reefs everywhere, One Tree Reef is an oasis in the desert that is the open tropical ocean. There is a richness, a diversity, and an abundance of life unrivaled in any other aquatic habitat, and certainly absent from the ocean surrounding the reef. There are over 850 different species of fish known to occur on One Tree Reef, and similarly large numbers of kinds of other animals and plants. Together, these plants and animals form a complex ecological system. Its members compete with one another, feed upon one another, and yet live together. Why are coral reefs so productive? Why do such large numbers of animals and plants occur together there? Can we understand and maybe one day effectively manage the intricately interrelated but abundant life found on reefs? It is in order to understand the workings of this complex biological system that the University of Sydney operates the One Tree Island field station. Scientists from the University of Sydney and from other institutions visit One Tree Reef to use it as their natural laboratory. Indeed, since One Tree Reef is now designated as a scientific zone within the Great Barrier Reef Marine Park, this reef cannot be visited by anyone other than scientists with permits to work there. It is a reef for science. Well, this is the One Tree Island field station. It's not much to look at, 3 huts on a beach. One of them is the home for the resident officer and his wife. The second provides accommodation for up to 8 scientists or postgraduate students. And the third is a combination workshop, storeroom, and a very primitive lab. Despite its appearance, One Tree is a very busy place, and its success as a research site over the past 12 years demonstrates that there are many aspects of coral reef science which can be successfully pursued for only a quite modest outlay of funds. Coral reef science, to be good science, doesn't have to be big science. Life at One Tree is simple. The evening meal, practically the only recreation, is a time for planning the next day's work, or for discussing some of the more fundamental questions about coral reefs. Unlike other environments, the physical structure of a reef has been built by the organisms which live there. The corals, in a myriad of forms and colors, and perhaps even more important, certain of the algae, secrete calcium carbonate, which over the ages has formed the rock structure which geologists know as the reef. One Tree Island and Reef would not exist except for the actions of these carbonate-secreting organisms. Large expanses of this coral-built rock are exposed at low tide. Scientists interested in coral reefs come to One Tree both because it's a reasonably accessible reef and because it's totally protected from all other kinds of human interference. The existence of accommodation and limited field equipment at the station also helps. The existence of the One Tree Island Station and others like it encourages the development of long-term, detailed studies at particular sites. Such studies are often experimental, and they may involve leaving recording equipment in place for months or even years. They result in important information on the temporal patterns in coral reef systems. As such, these stations are producing data which is an important complement to that on spatial variation, which is the natural outcome of ship-based research. This intertidal bench and the algae living on it are one of the subjects currently being investigated at the One Tree Island Station. For the last 2 years, a group of researchers headed by Dr. Tony Larkin have been studying nitrogen fixation over the whole of the reef.
The waters off One Tree Reef are very blue. They're also very lifeless. Very few fish, very few algae. And we believe that's due to the lack of nitrogen in the water. As you come onto One Tree Reef, you get green water with this great diversity of marine life that we see, the corals, the fish, and an abundance of the algae. We believe that's due to nitrogen fixation on all kinds of coral which we've got growing here. So we set up our program to study nitrogen fixation on the coral surfaces. The algae which fix the nitrogen are blue-green algae because they're blue-green, but generally they're microscopic, and on any coral where they live you can't see them. However, on some rocks they're very abundant, and you can actually see the blue-green smear of these algae all over the rock. And this would be fixing nitrogen extremely rapidly. In order to study the quantities of nitrogen produced by the algae, we chip off random samples of dead, algae-covered coral, take them back to the lab on One Tree Island. The coral surface is also home to various other creatures. What have we got here? Gas-tight jars filled with a measured amount of seawater are used to contain the algae-covered coral samples. Acetylene gas from a balloon is then injected into the airspace above the rock in each jar. The jars are then set out underwater in sunlight for several hours. Then a sample of the gas in each jar is removed for examination. If nitrogen fixation is active, some of the acetylene will have been converted to ethylene by the blue-green algae. A portable gas chromatograph is used to measure the amount of ethylene in each sample, and the results are registered on a chart recorder. These studies have shown that each year many tons of nitrogen are fixed. This flows into the reef waters and is passed on from organism to organism, a key process in supporting the huge diversity of life on this coral reef.
Another major line of research at One Tree concerns the organization of fish communities. As adults, reef fishes tend to be sedentary animals, and the various species present at one site are long-term neighbors of one another. Nearly all species possess a pelagic larval stage which drifts in the open ocean for days or weeks, depending on the species, before settling onto a reef. This fish, Acanthochromys, is a rare exception. It cares for its young and omits the pelagic phase. Here we are building traps or recruit collectors for these larvae. We're particularly interested in the process of settlement because the composition of local groups of fish ultimately depends upon which species happen to settle there. Settlement varies greatly, and a major aim for our research is to understand why. Sort of on top. Ooh, lovely. Supposed to come in. Yeah. The coral survives quite well, cemented onto its pipe. Placed on poles well above the sea floor and away from patch reefs, it is colonized only by newly settling recruits. The finely branched coral that we're using as recruit collectors seems to be attractive as a microhabitat for a large number of species. I've got some here that have been in the water for a while, so let's go down and have a look and see how they're working out. The young fish will settle among the branches of the collector, just as they will among the branches of that same coral where it occurs naturally on the surface of the reef. We can examine distribution of fish in these various natural habitats, but the recruit collectors permit direct measurement of settlement. At any site on the reef, we can determine the timing of settlement by each species of fish by clearing the recruit collectors of fish every few days. Here, the advantages of a collector over a natural coral head are clear. The collector is easily netted and can then be lifted and brought to the surface. Okay. The fish tend to wedge themselves into the coral, but they can be washed into the net. The collector can then be returned to its post.
So I'll bring up it.
collected fish are quickly preserved and returned to the lab for sorting. Results from these collector studies and from other studies of settlement by fish on natural reef habitats are helping us understand the reasons why certain species of fish occur in certain habitats, and why, even in those habitats, the number of settling larvae varies greatly from year to year. The island is inhospitable, often dry and salty. The way in which plants cope, and some thrive under these conditions, is another area of continuing research by scientists from the University of Sydney. This device, developed in the School of Biological Sciences, measures the flow of sap in a shoot. After the bark is removed, a small heater is attached to the sap-conducting wood. It is covered with foil to reduce the effect of the sun. Two thermocouples, one above and one below the heater, measure the sap temperature as it flows. The difference in these temperatures, indicated by the red band on the chart recorder, indicates the rate of sap flow. In a long-term growth experiment, tagged shoots are periodically measured on one plant able to tolerate very severe water stress. Other shoots of this yellow daisy, Melanthera biflora, are sampled at intervals and sent to the scientists in Sydney for analysis. The daisy uses salt to raise the osmotic pressure of its cell sap in order to get water from the very dry soil, and survives over most of One Tree Island. In 1843, the naturalist Jukes visited the island. From the sea, this large clump of pandanus palms may have been what caused him to give the island its name. Once he landed, he found inside the pandanus grove a strange structure which he referred to as a rude mass of dead sticks. In fact, the structure is a sea eagle's nest, and the same nest is present and occasionally visited by sea eagles even today. The island is occupied by a wide range of species of bird. Indeed, about 20% of the species of plants on One Tree Island were carried there by birds. In return, the plants provide several species of birds with nest sites and nest material. During the summer, most of the island is off limits, while the terns and other birds breed and raise their young. Over the years, these rookeries have continued to be successfully used, despite the increased human activity on the island. These small turnstones migrate thousands of kilometers annually between One Tree Island and Siberia. While some birds feed on the island, most feed over the reef, and on return to the island, their droppings provide the nutrients required by the plants. Boobies often dive for fish on the reef edge. One Tree is a peaceful place, but for Graham and Wendy Russell, who manage the station, it is no Shangri-La. They both have a diversity of jobs to do if the station is to function properly.
We're basically on a 24-hour call here for any assistance is needed by anyone. But apart from that, we figure out our own work schedule, maintenance schedule on the island.
Oddly, for an outpost many kilometers off the coast, regular and emergency communication is made via the Flying Doctor radio base in Charleville in inland Queensland, over 600 kilometers away.
00-692-3600.
Gillian Eckert is a PhD student currently using One Tree Reef for her research.
The fish I'm working on, Thylassoma lanare, has a few interesting characteristics. In any group of fish, all of them are female except the largest one, which is a dominant male. And if I remove the male, then the largest female actually changes sex and becomes a male. And to do this work, I spend a lot of time in the field, actually in the water, doing observations. And I'm using this piece of equipment at the moment, which is attached to an underwater tape recorder. Larval fish arrive at One Tree Reef over the summer months, here sharing the coral with a sea cucumber. The preferences of each type of fish are being studied by setting out different types of coral at varying depths. At regular intervals, the squares are examined and the species of fish settling on each are recorded. Some fish species may have a preference for a particular type of coral, and for the rest of their lives they will remain near the same piece of coral that they first settled in. In another part of the lagoon, I'm researching the phenomenon of sex changes in reef fish. Only 10 years ago this was thought to be unusual, but recent research has shown this to be quite common. On this isolated patch reef, I'm looking in detail at the moon wrasse, Thylassoma lanare. Where there are small groups of wrasse, the largest is always a male, while the rest are females. The male tends to be brightly coloured and is very aggressive towards the others. In this experiment, the male is being removed to find out how long it will take the largest female to become male. Underwater fishing with a small hook works well for this because the aggressive male usually rushes in and takes the bait first. Within a couple of weeks, a female will have changed sex to take the male's place and will spawn with the females. Anemone fish also change sex, but in their case, the largest fish is always a female and the smaller ones are males. Fish are taken back to the island to be weighed and measured. The gonads are also removed to check the sex of the fish. Compared to many commercial fish species, the gonads are quite small. This is because the moon wrasse spawns only a few hundred eggs every day throughout the summer, rather than a huge number all at once. There is still a very great deal of research needed to understand the biology of even the most common of the fishes that inhabit One Tree Reef.
Some of the most important of marine plants at One Tree Reef are the symbiotic ones, which exist as single cells within the tissues of animals such as the corals. One such symbiotic alga occurs within cells of a common finger-like sponge found throughout the lagoon. This alga is the subject of study by another Sydney postgraduate student, Françoise Pironnet. Like other symbiotic algae, its photosynthetic activity produces organic matter which is used directly by the sponge host. To measure the rate of photosynthesis of the organism, the sponge is held inside a sealed chamber with an oxygen electrode. When the chamber is illuminated, photosynthesis begins and oxygen is released. The oxygen is then detected by the electrode and the electrical signal recorded on a chart recorder. Samples of the sponge are also analyzed using a spectrophotometer. These sponges are not eaten by fish because of anti-feeding compounds within them, which make them toxic and distasteful. This research may isolate these compounds and discover how they are formed. Despite its small size and primitive facilities, the field station at One Tree Reef has been successfully used by many scientists over the past 12 years. They've provided a wealth of information about this reef, information with important general implications for our understanding of coral reef systems. The information provided from the work done at One Tree is out of all proportion to the small size of the reef, the island, and the station itself. Yet, while we know a lot about One Tree Reef, there is still a lot more that we do not know. Understanding and management of Australia's Great Barrier Reef has been, and in the future will continue to be served by research being done here at One Tree.